So we know our plasma membrane acts as a barrier. So therefore, we know molecules can't just simply enter inside of the cell because we have this plasma membrane barrier. However, in certain situations, we need molecules to enter inside of the cell. So what are the ways that molecules enter inside of the cell? Well, one way is through simple diffusion. So simple diffusion is when a molecule simply just diffuses straight through the barrier. So what kind of molecules can go through simple diffusion where they simply just cross straight through the plasma membrane barrier? Well, small nonpolar molecules and also gases can go through simple diffusion where they simply cross straight through the plasma membrane barrier. So why do small nonpolar molecules simply diffuse straight through this plasma membrane barrier? Well, remember what this plasma membrane is. It's this phospholipid bilayer membrane. So we know these phospholipids have these hydrophobic tails, so we know the interior, the core of this plasma membrane is nonpolar. So we know the core of this plasma membrane is hydrophobic and nonpolar. So all throughout this plasma membrane, in this inner core, are these hydrophobic tails, these nonpolar tails. So therefore, we know our rule, likes dissolve likes. So therefore, we know nonpolar molecules, for example, small nonpolar molecules, can simply dissolve and diffuse in this nonpolar internal region. So therefore, if they can dissolve in this nonpolar region, they can simply dissolve and diffuse and cross straight through this barrier membrane. However, if we have a polar molecule, for example, a polar molecule or a charged molecule, it cannot dissolve in this nonpolar core. So these polar molecules cannot dissolve in this nonpolar core, so they can't cross this barrier. So again, simple diffusion is when molecules just simply diffuse straight through this barrier. This barrier is essentially invisible to them and they can simply go through. And also small molecules can squeeze in between these spaces. But something important to realize is this is the exception. Only very few molecules, only gases and very small nonpolar molecules can simply diffuse inside the cell. Generally speaking, this plasma membrane does act as a barrier and most molecules cannot cross through. However, small nonpolar molecules can through simple diffusion. So that's one example. Another example of ways molecules enter inside of the cell is through facilitated diffusion. So again, remember we have this plasma membrane, this bilayer membrane, but sometimes we have proteins in this plasma membrane. For example, sometimes we have these protein channels. And what these protein channels do is they create these little holes. They create these little holes in this plasma membrane. So therefore, once these protein channels create these little holes, we know certain molecules can cross straight through using these channels. So this is referred to as facilitated diffusion because this diffusion is facilitated with the help of these proteins, for example, these channels. But something important to realize are all protein channels can be characterized by the way they're regulated and by which ion they're specific for. So what do I mean? So let's say we have this channel. We know all channels can be characterized by the way they're regulated and specific for. So let's say this channel is regulated. Let's say this channel happens to be regulated by ligands, which essentially means they're regulated by molecules. Ligands are essentially molecules. So what does that mean? That means this channel is normally closed. However, if the correct ligand binds, then this channel opens. So now it opens. So again, when the ligand binds, it opens. When the ligand is not bound, it's closed. So therefore, we can see it's this ligand that regulates whether this channel is open or not. But let's say the ligand binds and opens the channel. Now what? Well, again, we know all channels can be characterized by what ion they're specific for. So let's say this channel happens to be specific for sodium ions. So therefore, once this ligand binds and this channel opens, then sodium ions and only sodium ions can flow through. Because again, this channel is specific for sodium ions. And something important to realize is once this channel opens, sodium can only flow with its gradient. For example, if we have a lot of sodium outside of the cell, if we have a lot of sodium outside of the cell and a little bit of sodium inside of the cell, then when this channel opens, sodium is going to flow inside of the cell. It's going to flow with its gradient. So again, but something important to realize is potassium will not be able to enter because this channel is specific for sodium ions. So it's going to create a hole that's exactly the right side for sodium ions and only sodium ions. But so potassium is too large, so it's not going to be able to flow. So again, all channels can be characterized by the way they're regulated and which ion they're specific for. So let's do another example. Let's say we have this completely different channel. And again, all channels can be characterized by the way they're regulated and which ion they're specific for. So let's say this channel is regulated by voltage. So that's interesting. What does that mean? Well, we know normally inside of the cell, the voltage is at negative 70 millivolts. So when the voltage is at negative 70 millivolts, the channel is closed. However, if the channel increases to negative 45 millivolts, then the channel opens. So at negative 45 millivolts or above, the channel opens. At negative 70, the channel is closed. So clearly we can see it's the voltage that regulates whether the channel is open or not. So how does this work? Well, we know voltages create these electric fields. And again, remember, this is a protein channel. So it's a protein channel made out of amino acids. And certain amino acids are charged. For example, lysine and arginine and certain amino acids are charged. So therefore, if we create the right voltage, it'll create the right electric field that can exert forces on these amino acids. And when this electric field exerts forces on these amino acids, it goes through a conformational change where it opens. So at certain voltages, it doesn't apply enough electric field, but at other voltages, it applies the exact right electric field to apply the exact right force to open this channel. So we see it's this voltage that regulates whether this channel is open or not. But let's say we have the right voltage, and let's say the channel opens. Now what? Well, let's say this channel is specific for potassium ions. So once this channel opens, we know potassium ions are going to flow, and we know they're going to flow with their gradient. And potassium and only potassium ions will be able to flow. You might wonder, what about sodium ions? They're smaller than potassium, so wouldn't sodium ions be able to flow? Well, it's a little complex, but the point is, sodium won't be able to form enthalpically favorable bonds to allow it to go through this region and enter. Only potassium is the exact right size and charge to form the enthalpically favorable bonds to allow it to bond to this region and then flow straight through. But the point is, once this channel opens, we know potassium ions are going to shoot down their gradient. So again, all channels can be characterized by the way they're regulated and which ion they're specific for. And something important to realize are channels usually are specific for ions. It's ions like sodium and certain electrolytes that can flow through these channels. So that's channels, but we can also have carriers. So carriers are another type of facilitated diffusion. So how do these carriers work? Well, let's draw it a little bigger. Essentially what happens is, let's say we have a glucose carrier. So how does this work? Well, we have this glucose concentration gradient with a lot of glucose outside, a little glucose inside. So we know glucose wants to fall down its gradient, so it wants to enter inside of the cell. So how does it do this? Well, first, it binds to its carrier. So first it binds to its carrier. Once it's bounded to the carrier goes through a conformational change. When it goes through a conformational change, now it can release that glucose, and now that glucose enters inside of the cell. So this is through carriers. And notice the difference between channels. We know with channels, they create these little holes. So we know the ions can just shoot down their gradient, very quickly shoot down their gradient. However, these carriers and transporters are a little different. The glucose needs to bind, goes through conformational change, and then enter. And again, channels are specific for ions, while carriers can be small molecules and ions. But something else important to realize is every single example we've done so far, the molecules went with their gradient. For example, we had this glucose gradient, so the glucose went with its gradient. However, these carriers and transporters are unique because we can have these active carriers or transporters, which are sometimes referred to as pumps, but these active carriers and transporters can use ATP to push molecules against their gradient. So this is the only technique that the cell can use to push ions or molecules against their gradient using active carrier transporters, which use ATP to push molecules against their gradient. So how does this work? Well, first, we know glucose wants to go with its gradient in this direction. However, we can use an active transporter to push glucose against its gradient to create a concentration gradient. So how does this work? First, glucose binds. So first, glucose will bind to this carrier. Then ATP will bind. Now, once ATP binds, now we can hydrolyze this ATP, using up the molecule of this ATP to cause a conformational change to now push glucose against its gradient. So notice we used ATP to push glucose against its gradient. So again, really quickly again, first, glucose binds. So first glucose binds, then we use ATP and hydrolyze that ATP to essentially push glucose against its gradient. And then this phosphate that was still covalently attached, well, now we can hydrolyze that, bringing the carrier back to its original situation. And now we can do the whole process over again. Now a new glucose molecule can bind, then ATP gets hydrolyzed, pushing that glucose against its gradient. So this is the only way the cell can use to create these concentration gradients. We're creating a concentration gradient using ATP. So the most famous transporter pump that you'll see that uses ATP to create these gradients is the sodium-potassium transporter. And nearly every single cell in your body has these sodium-potassium transporters. So how does this transporter work? Well, first sodium binds. So first sodium inside of the cell binds to the transporter. Then once it binds to the transporter, now ATP binds to the transporter. Now ATP is hydrolyzed. It's hydrolyzed with this phosphate still bonded, and now the sodium leaves. So now we've essentially pushed sodium outside of the cell. Now potassium can bind. Now an external potassium can bind. So now potassium binds. Now this phosphate is hydrolyzed. And when the phosphate is hydrolyzed, we go through a conformational change, and now potassium enters inside of the cell. So notice what we've done. We've used the energy in ATP to push sodium ions outside of the cell and push potassium ions inside of the cell. So this is a way to use the energy in ATP to create the sodium and potassium gradient. But why do we do this? Why do nearly all cells in your body use the precious ATP, uses the energy in these precious ATP molecules to create these concentration gradients where we have a lot of sodium outside of the cell and a lot of potassium inside of the cell? Well, now we have a sodium gradient, and we can use the sodium gradient because we know with the sodium gradient, the sodium desperately wants to enter inside of the cell. If we keep on pushing sodium outside of the cell, sodium is going to want to enter inside of the cell. So we can use that. We can use that energy of sodium wanting to enter inside of the cell to do stuff. For example, let's say we have this molecule X. It doesn't matter what it is. Let's say we just have this generic molecule X, and let's say for whatever reason, the cell wants to take that molecule X and bring it inside of the cell. How can the cell take this molecule X and bring it inside of the cell? Well, as long as we have the right transporter that has the correct structure, it can essentially use the sodium gradient because we know sodium desperately wants to enter inside of the cell. So this transporter can utilize that. It can exploit that and utilize the sodium wanting to enter inside of the cell, and it can use that sodium entering inside of the cell and use that energy to take this compound X and bring it inside of the cell. The point is, it's using the sodium, the energy in the sodium wanting to enter inside of the cell. It's using that to push this compound X inside of the cell, and it can do that as long as we have the right transporter. So again, the way this works is first, the sodium and the compound X will both bind to the transporter. So first they'll both bind, then the transporter will go through a conformational change. Now the molecules will be released. So notice what we've done. We've essentially used that sodium gradient to push that compound X inside of the cell. So when we do this is referred to as a symporter. This would be a symporter because both the molecules are going in the same direction. But let's do another example. Again, let's say for whatever reason, we want to take this compound Y and we want to push it outside of the cell. How can we take this compound Y and push it outside of the cell? Well, remember, this sodium strongly wants to enter inside of the cell. So we can use that sodium entering inside of the cell and use that energy to push this Y outside of the cell as long as we have the right carrier with the correct conformation and the correct structure. So first the sodium will bind. Then the transporter will go through a conformational change. Now the sodium will enter. So the sodium has entered. Now that it's done that, now the compound Y can bind. So now the compound Y binds, now goes through a conformational change. Now the Y leaves. So we were using that energy and that sodium entering, using that energy to push this Y out. So this transporter would be referred to as an antiporter, because these antiporters is when the molecules go in the opposite directions. And we can also have uniporters where it's only one molecule binds and then enters. But the point is, we use that sodium-potassium transporter to create the sodium gradient, and once we have the sodium gradient, we can use the energy in that gradient to move molecules around. And just some terminology, is when we have a channel or carrier, when we have a carrier that uses ATP directly to push stuff against its gradient. That's referred to as primary active transporter. So this is a primary active transporter because it directly uses ATP to create the sodium gradient. However, if we have a transporter that uses one concentration gradient to push another molecule down a gradient, that's referred to as secondary active transporter. So this would be a secondary active transporter. So if ATP is the source of energy to move a molecule, that's a primary active transporter. However, if we're using another concentration gradient to move another molecule, that's secondary active transporter because the energy is coming from this concentration gradient. So then we move this Y molecule. Now if we had another transporter that used this Y molecule gradient to push another molecule, that would be a tertiary transporter. But again, this is the way we move molecules around in the cell.